Hello, I'm Professor Sims, and this video is about bacterial structure, simple stains, negative stains, Grimm, and acid-fast stains. This is the fourth of a series of 10 lab sessions held as part of my Laboratory for the Fundamentals of Microbiology course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include learning to prepare microbial smears, simple stains, negative stains, Gram and acid-fast stains, and becoming competent at observing stained specimen. You will observe and describe bacterial cell morphologies, and of course, you want to make sure that you understand the safety rules and disposal procedures for these experiments. Preparing samples for viewing with a microscope depends on how you do it, depends on what you are looking at and what it is that you're looking for. For this class, cells may need to be fixed to a slide, dyed with stains, or you may have tissues that can be cut into thin layers in order to view those. Labs 4 and 5 will focus on 7 specific techniques used for preparing samples for viewing, all of which are used for very specific purposes. Simple and negative staining is used to determine cell morphology. Gram and acid-fast and also endospore and capsule stains are used to identify structural elements of bacterial cells, like their cell wall components or their virulence factors. Flagella staining allows for visualization of flagella and identification via observed flagellar arrangements. In the last lab, Lab 3, you may have had some trouble viewing some of the microbes, and one of the reasons for that is because cytoplasm is essentially transparent, and that makes viewing with a light microscope very difficult. In order to observe things like your morphology, structures, extracellular characteristics, we must have a way to increase the contrast of the cells. And the way that this is done is by using microbial stains. Most stains work by binding to cellular components like proteins or nucleic acids, and they do this utilizing an ionic charge. An ionic charge, like positive or negative, has to do with how many electrons are in the outer membrane. The outer cell membranes of most cells have an overall negative charge, so stains known as basic stains. These stains have a positive charge, and because opposites attract, the positive charge of this basic stain is attracted to the negative charge of the cell membrane. Stains are made up of chromogen plus an oxochrome. So chromogen is what gives it its color, okay? So chromogen is a benzene ring and a chromophore, which imparts the color. And then the oxochrome gives the stain its charge. Now, it is important here, I want to make sure to point this out, that benzene is a potential carcinogen. So when you are handling these dyes, these stains, you want to be very careful. Handle them with great care. In the last slide, I did mention a basic stain. A basic stain is essentially, it's like paint. It paints the cells. That is, the cells retain the color while the background stays clear. And this works because a basic stain has a positive charge that is attracted to this negatively charged cell membrane. Example of some basic dyes, basic stains include methylene blue, safranin, crystal violet, carbofuchsin, malachite green, and there's a few others like brilliant green K, a couple of others that you will see in upcoming labs. Basic dyes are used in most of our staining techniques. So your simple stains use them, Gram stains, acid-fast, endospores, and even flagellar stains use basic dyes. Now, on the whole opposite side of that is your acidic stain. An acidic stain is negatively charged. So acidic stains are actually repelled by the cells, and this means that the dye does not impart color to the cells, but dyes the rest of the slide, the background, instead. And then your cells stay clear. So you can see how the background here is dark, and then these in white here, these are the cells. Examples of acidic stains are eosin, nigrosine, and India ink. Acidic dyes are only going to be used in this class anyway for the negative stain and the capsule stain. It is important to learn what cell morphologies are, and make sure that you are aware that cell morphologies are very different from colony morphologies. Colony morphologies is what we've seen in labs 1 and 2, where you're describing how a colony of genetically identical cells that have all been aggregated to form a mound of cells that you can view with the naked eye. That's a colony morphology. And they have lots of different descriptors like size, color, margin, elevation, all of that stuff. Cell morphologies are a lot more specific and they're a lot more difficult to observe because now you're not looking at a huge aggregation of cells. Now you want to look at individual cells, right? So the first thing you're looking for is shape, and then the second part we'll talk about in a minute is arrangement. Cell morphology is only going to be your shape and arrangement. There are more shapes than this that are shown here, but these are the most common and these are the ones that you're going to see in this class. You have coccus or cocci, which is plural, bacillus or bacilli, plural, and spirillum or spirilia, plural. In order for a cell shape to be classified as cocci, it has to be a perfect sphere. It can't be an oval. It's not going to be like irregularly shaped. It has to be a perfect circle. In order for something to be classified as bacilli, in general, bacilli are rod-shaped, but they can look quite different. They can be kind of long and skinny rods. They can be a little short, squat-looking rods. And these, a lot of the times, are misidentified as cocci, so you want to be really careful. But a whole lot of bacilli are right in the middle. They're not super thick or thin. They're not super short or long. They're kind of pill-shaped like these guys. And then spirillum, don't confuse these for the spirochetes. The spirochetes spiral around so much that they look like springs. Spirilia, not so much. Spirilia are often miscategorized as bacilli. What you're looking for is basically this tilde shape. It goes up, it goes down, it goes up, it goes down again. That's spirilia. As for arrangement, again, you have other varieties, lots of other varieties, but these are the most common and these are the ones you're most likely to see in our class. First of all, you do have cells that occur individually, and they would have the same name as their shape. So if you have cocci that are almost always appearing by themselves, then that would just be called cocci. It's not monococci. If the cells are appearing individually, their arrangement is the same as their shape. If they're occurring in pairs, like these here, then that arrangement is diplo. If they're appearing in chains of 3 or more cells, then that is strepto. And if they're in clusters that kind of look like grapes and they're all on the same plane, in other words, these cells are not laying on top of each other vertically, but they're all connected to each other horizontally and they look like little clusters of grapes, that is staphylo. So how are these stains done? Well, first, you've got to prepare the cells to be stained. And while you can do a simple stain on a wet mount slide, in fact, you will be doing this for experiment 1, before doing any of the other types of staining techniques, cells must first be affixed to the slide via dry mount, which is also known as a smear. So first you would start with a new clean microscope slide and you're going to label it. Then you're going to apply a loop full of broth, or if you're not using a broth you're taking from a plate, you would add a small drop of water and then add your cells to the water. And of course you're doing this aseptically, so you would want to be doing this with a sterile loop. You would spread the cells evenly on the slide in a circle about the size of a dime, and it does help to use a Sharpie. You can take a Sharpie and draw on the underside of the slide a little circle about the size of a dime, and that helps with keeping your smear a good size and making sure your cells are evenly spread within that little circle. But it also, having that Sharpie line also helps you when you're trying to view the cells, because you can view the Sharpie line first because it's really, really big compared to your cells. And you can get that into focus, and then that puts you in the ballpark of where you need to be looking for your cells in order to focus your cells. Another thing to consider with your smear is you don't want the cells to be on there in a layer that's too thick or too thin. A smear that's too thick will result in an uneven stain, and it may also lead to false results for your differential stains. And then a smear that's too thin will result in cells that are really hard to find, and they'll be difficult to view. And they can also mess with the results of your differential stains. So a good bacterial smear will be thinly visible to the naked eye, and it will look much more like this slide here on the left than this. This is way, way too many cells. It's going to be way too thick, and it's going to be really hard to stain. After a smear is prepared, most stains require the cells to be heat fixed. Heat fixing prevents the specimen from washing off the slide during the stain procedure. It also inactivates any enzymes present. It prevents the cells from undergoing autolysis. It also kills the organism. So if you are trying to view, like in lab 3, you're trying to view specimens that are swimming around, you wouldn't want to use this technique because it will kill your swimmers. Heat fixing requires holding a slide with the clothespin, and the reason you use the clothespin is because the wood from the clothespin doesn't conduct heat as well as the glass from the slide, so that prevents you from burning your fingers. And you have to apply direct heat to the slide until the liquid on the slide evaporates. Now, we will not be heat fixing. You guys are not going to do the heat fixing, but you still need to know how it's done, and you still need to know why it's done. For experiment 1, you're going to do 2 simple stains. You'll be doing this on slides that have already been prepared. So the bacterial smears were made and the cells have been heat fixed. Then all you have to do is add the stain, time it for a minute, and then rinse it, blot it dry, and you can view it. You want to observe these slides, record your results in the lab report template. For experiment 2, you're not going to have the acidic dye to use, so you're going to learn how a negative stain is performed by watching this video. While you're watching the video, you want to make sure that you make notes of all of the procedural steps, and those actually are shown here as well, but also all of the equipment that is used to perform the stain. And you'll need this information in order to answer the guided questions in your report template. Simple and negative stains, these both require only one dye, and it's essentially one step. But differential stains are going to require multiple steps and they're used in order to separate bacteria into different groups based on what they look like after all of the steps are complete. All differential staining procedures have these same 4 steps. You have the primary stain, which is the first stain that colors all of the cells. The mordant, which augments binding of the primary stain. In other words, the mordant helps the cells retain the primary stain if they have the structure that you're looking for. The next step is the decolorizer. The decolorizing step removes the primary stain selectively based on the presence or absence of a specific characteristic. So if the cells have the structure that you're looking for, the mordant will hold the primary stain when you're decolorizing. But if the cells do not have that structure, then the primary stain gets washed away with the decolorizer. And then the counter stain is a contrasting color, so it's going to be a different color from the primary stain. The counter stain will stain cells that were decolorized in the decolorizing step. So for all differential stains, if the cells have retained the primary stain, that is a positive result. It means it does have the structure that you were looking for. If they do not retain the primary stain and instead they retain the counter stain, then that is a negative result, meaning it does not have the structure that you were looking for. The gram stain is a differential stain that is usually the first step in identifying an unknown species of bacteria. Gram-positive cells have larger amounts of peptidoglycan and a single cytoplasmic membrane that also has teichoic acid in it. Gram-negative cells have less peptidoglycan, they have 2 cell membranes, and they have lipopolysaccharides. So this figure here on the left, 1 cell membrane, and you have this really large outer layer that has peptidoglycan, proteins, and teichoic acid. That is the structure of cells that are classified as gram-positive. Over here, you have 2 cell membranes, and within those cell membranes, you have proteins again, but you also have these lipopolysaccharides. And then sandwiched between those 2 cell membranes is a much smaller layer of peptidoglycan. Cells that have this structure are classified as gram-negative. So when you do this stain, you're determining if the cell walls have this structure or if they have this structure. And that helps you to differentiate a bacterial species into two big groups. It essentially divides the possible species it could be in half. So these are the steps. This is what they look like. In gram stains, the primary stain is crystal violet, which colors the cells purple, no matter if they're gram-positive or negative. Essentially, the first step is just a simple stain, right? It's using crystal violet, which is a basic dye, to color the cells. Then you have the mordant. The mordant is iodine. Iodine binds to the cell wall, and it will form a crystal violet iodine complex if that thick layer of peptidoglycan is there. If that thick layer of peptidoglycan is there, the iodine will anchor the crystal violet to that peptidoglycan. The decolorizer is 95% ethanol. So if that large peptidoglycan layer is absent, the decolorizer will remove the crystal violet from the cells. And then the counterstain safranin is used because if the cells are gram negative, after the decolorizing step, the cells will be clear and they'll be hard to view unless you stain them. So the counterstain, if the cells are gram positive, they're going to retain the primary stain and they won't take up the safranin, and they'll be purple when you view them. If the cells are gram negative, then they should be pink when you view them. Now for experiment 3, you're going to do the gram stain procedure using slides that have already been prepared and heat fixed. After you finish the gram stain, you're going to observe the slides. You're going to look for the gram reaction based on the color, and also you're going to be looking for the shape and the arrangement of the cells. And these pictures here are representative of what you should see on those slides. You should see purple Staphylococcus cells. This would be gram-positive Staphylococcus. And you should see pink bacilli, gram-negative bacilli. In general, an atypical gram stain can result if you have a specimen that's old, if your smear is too thick or too thin, if you have under or over decolorization or under or over heat fixing. These two here are very common, and you should absolutely know how these errors would affect the results of your gram stain. So if you over decolorize a gram stain, you can have a false negative result. If you under decolorize a gram stain, you could have a false positive result, and that is very, very common for both of those. And then also, if you over heat fix cells, then what happens is the cells get cooked and they turn like a dark brown or black color, which means that they won't have that purple or pink color. They'll be something else, right? They also kind of shrivel up so that you can't determine their shape and their arrangement. And then if you under heat fix, what happens is the cells just get washed off the slide when you're adding the stain or rinsing, and you won't be able to see them. And then also something that's not listed here is you can have contamination. So if you're gram staining a pure culture, you know it's a pure culture, but you have two different color cells and you have two different shapes and arrangements, then you probably have two different species on that slide. And if that's not supposed to be two species, then you probably have contamination. And then you have another consideration is that some organisms simply do not reliably gram stain. And a classic example of that is the genus Mycobacterium. Mycobacterium don't reliably gram stain because of their mycolic acid layer. Mycolic acids are non-permeable, waxy substances present in the cell wall of acid-fast bacteria. They increase virulence by protecting the cells from desiccation, which is basically dehydration. They protect them from phagocytosis and even antibiotic treatment in some cases. So here is structure of Mycobacterium. And if you just looked from this part down, it almost looks like a gram-positive structure with one cell membrane and then a layer of peptidoglycan and succoic acid on the outside. But you have this layer here of mycolic acids. And these mycolic acids make it difficult to do a gram stain because they kind of block the stain from getting to the cell wall. So in order to identify this genus of bacteria, an entirely new staining technique was developed called the acid-fast stain. But why? Why was it so important to find a way to identify Mycobacterium? Well, that's because this genus of bacteria causes some of the worst diseases in human history. We've got here tuberculosis, which is caused by Mycobacterium tuberculosis, and then of course leprosy, caused by Mycobacterium leprae. In acid-fast staining, the primary stain, carbylfuchsin, is added to the cells while they are being gently heated via steam heat. The heat acts as the mordant because it drives the primary stain past the mycolic acids, if they're there, and into the cell membrane. Then the cells are removed from heat and the decolorizer acid alcohol is added. If there's no mycolic acids present, then the cells will become clear at this stage. But if the mycolic acids are present, the primary stain will be trapped by the mycolic acids and they remain attached to the cell wall after the decolorizing step. And then the counterstain, brilliant green, stains the decolorized cells a teal bluish green color. At the end of the acid-fast stain, acid-fast positive cells will have a reddish color from the carbylfuchsin, and acid-fast negative or non-acid-fast cells will have a bluish green color from the brilliant green. I also have seen some procedures that use methylene blue as the counterstain, which is perfectly acceptable as well. So in general, acid-fast positive cells will have a reddish color, and acid-fast negative will have a blue or bluish green color. For experiment 4, you will learn how an acid-fast stain is performed by watching this video here. You want to be sure that you make notes of all of the procedural steps, any and all equipment that's used, any results that you get, because you're going to need this information in order to answer the guided questions in the report template. And here I've put a couple of examples. Both of these are mixed cultures, so there's both positive and negative. So you see this is kind of a reddish purplish color. This is more of a reddish orangish color, but either one of those is considered acid-fast. It's positive for acid-fast. This one here used the methylene blue as the counter stain, so the non-acid-fast are much bluer than they are here. These ones are more kind of a blue-green teal color, and that's because the counter stain for these was the brilliant green. Okay, but you see in both cases, the more reddish colored ones are positive for acid-fast, and the more bluish colored cells are negative. For this lab, it's going to be really important to follow safety procedures. So as always, you want to disinfect before and after doing your procedures. PPE is highly recommended. And make sure you carefully read all of the procedures. Treat all specimens as potential pathogens. But for this one, it's really important. Remember that those stains contain benzene. So you want to be very careful with them. You want to try not to spill them. Try not to get that stuff on your hands because benzene is a potential carcinogen, cancer-causing agent. Also, be careful with your glass slides. You don't want to break those, cut yourself. We talked about this in the intro lab. And for your observations and interpretations, so like I said, you want to make your observations and answer all of your report to template guided questions. Make sure you understand why the cover slip was necessary for the wet mount, but not for any of the other slides. Are you able to observe and describe bacterial cell morphologies? Do you understand how all of the stains work? Were you able to differentiate gram-positive and gram-negative cells? Do you know the potential errors in those procedures? Were you able to differentiate acid-fast and non-acid-fast cells? Thank you guys for watching. Don't forget to do the reading. Check the description below for more videos related to these topics and leave your questions for me in the comments below.